Okay, good morning, everybody. Welcome back to human biology. And there's just a couple of things I need to tell you about. First of all, please remember that you have a quiz on Wednesday. That quiz will be 25 questions. You'll have a half an hour to complete the quiz. That means you have to work fast. This is a closed book exam. Do not attempt to cheat by using your book or your notes or whatever, because you'll soon find that you're going to fall behind and you will not be able to complete the quiz. Okay, the answers must be up here in your head. It must be clear in your head, and if you do so, you will be able to answer them quickly. Now, one word of advice I always give to people for doing multiple choice, doesn't only apply in this class, it applies for doing multiple choice in general. That is, when you read a multiple choice question, before you look at the answers, have an answer in your head. In other words, just read the question and answer it as best you can. Then seek that answer in the choices. Do not read the question and then frantically try and pick an answer. Have the answer in your head before you attempt to choose an answer. Worse still, don't read the answers and then read the question, which people do. You just confuse yourselves when you do that. Instead, have an answer clear in your head, then look for the answer in the answer list that most closely fits to what you have in your head. That's the first thing. The second thing, the last time I did a recording, I realized afterwards, looking at the recording, that when I access videos and things on YouTube, whatever, that unfortunately those are visible to me, but they're not visible to you. For some reason, the screen sharing does not pick those up. Please, wherever you see one of those video links in your PowerPoint notes, please follow it up, because those little videos, very often they will provide you with a really clear explanation of things, make it much more easily understandable than all of my bloviating about it. You get a real good idea from the notes, from what I say, etc., etc. But often that visualization is what really key to kind of understanding with the processes. That applies especially today, is what we're going to talk about. Please follow those video links and make sure that you that you view them. I also do put in video links for things that are just for interest's sake, and that I always make clear to you, this is just for your interest, just to spark your interest, or because it's pretty or whatever. Okay, so we've been talking about the cell and about cells in general, really realizing that we are multicellular organisms. We are eukaryotes, and we consist of a vast number of cells performing all sorts of different functions. And we learn something about the specialization of those functions. Cells specialize by turning off certain genes and by turning on certain genes. And by doing so, they acquire their different identity. We use the example of nerve cells and skin cells. When we look at nerve cells and skin cells, they contain exactly the same DNA, exactly the same DNA. But in one cell, certain genes have been turned on, others have been turned off, and in the other one, other genes have been turned off and others have been turned on to give those cells their individual function. Now, there's something else which is locked into all the genome of all cells, and that is that all cells have a defined lifespan. That lifespan is very, very different from one kind of cell to another. So, for example, nerve cells are extremely long-lived. In fact, they may last for your entire lifetime. Other thing I've told you, skin cells are very short-lived. They reproduce the whole time to replace themselves. Very, very short. The exact reasons why certain cells have a certain lifespan, we don't always understand, and we don't always understand how the cell measures its lifespan, but they do. And when a cell dies, it doesn't die chaotically. It doesn't like scream and roll on its back and burst or whatever. The cells die in an orderly fashion. They are programmed. When they are required to die, they will shut down various functions one after the other, and the cell will kind of wither away. Many of its components will be recycled into the organism, but the cell will die in an orderly fashion. They sometimes call it self-suicide. And its proper name is apoptosis. Apoptosis is actually Latin for leaf fall, for what's starting to happen now outside us, when the leaves die and fall off. That is apoptosis. And apoptosis in our cells is orderly cell death. I'll give you a simple example. It's a key process in development. And so, for example, our hands and our feet in the uterus, they develop as a paddle, a solid piece of flesh like that. And during development, the fingers or toes, whatever, they develop. And this section here is initially joined to all the others. And then these cells die by apoptosis, and they die to liberate the fingers. So that's a simple example of programmed cell death. But it occurs throughout our body the whole time, depending on lifespans differ, but it is an essential part of regulation of the body. It can go wrong. And that's the next thing that I want to tell you about. Because when these systems of cell organization start to go wrong, we can end up in real trouble. And when the orderly apoptosis, the orderly development of the cells is removed, then the normal tissue structures are lost. That is usually due to two causes, and the one is mutation, that is changes in the DNA of the organism, changes in its genome, but the other is infection, and usually infection by a virus of some sort. So it leads to these disorders of growth and the cell division cycle and things like that, and produces very often what we refer to as a tumor. That is a group of cells which stand out of the normal organization structure. Some tumors are completely benign. For example, a wart. Warts are very common. They're caused by a virus called the papillomavirus, and they are normally completely benign. There are circumstances where they can be dangerous, but normally a wart is completely benign, and it's not a danger, because although it's disorganized, that's what's happened in a wart. The skin cells have become disorganized. They overproduce skin cells. They overproduce cells which have a lot of keratin to them. So it produces that typical warty look. But a wart is not dangerous for one important reason, and that is the cells preserve their lifespan. The cells of a wart still have a lifespan which is finite, and they will die eventually. It means that your body has a chance, your immune system eventually will catch up to the wart and be able to eliminate it. It becomes extremely dangerous when cells lose that inbuilt clock. When cells lose their mortality and become immortal, we enter into a very, very dangerous situation, and that is what we classically call cancer. Cancer cells typically are immortal. They fail to apoptose, and they reproduce out of control. And there are two kinds of cancer. The one is called a benign, the other is a metastatic. Benign cancers, benign makes it sound like it's harmless. Benign cancers are not harmless. They're often extremely dangerous, but they are circumscribed. They usually grow just as a single unit, and although they can actually become very large, they around them they will have a connective tissue layer usually. So they are much more discreet. In many circumstances, if that occurs in the body somewhere, in abdomen or wherever, they can be removed without a lot of difficulty. But there is one circumstance where they are extremely dangerous, and that is when they occur on the brain. And many brain cancers are in fact benign. They are benign tumors. That doesn't stop them from being dangerous or even deadly, as you'll realize. The opposite of benign cancer is a metastatic cancer. And a metastatic cancer is not so well circumscribed. And it has a tendency for cells to break off from it and to migrate to distant parts of the body. So metastatic cancer is continually producing clones of itself. Those cells that drift off the initial tumor, called the primary tumor, can be taken to many parts of the body where they will begin to divide. They divide out of control and produce metastatic cancers from the primary cancer. It is the responsibility of the immune system. We produce potentially tumorous cells the whole time. They are commonly produced during the process of cell division. But our immune system has very, very skillful ways of assessing cells and telling whether or not they are likely to be tumorous. And our immune system does a very, very good job, usually, of wiping out potentially tumorous cells. But obviously, cancer is a major illness in human beings, a major source of human mortality. So there are those which evade the immune response. But just remember, most cancers, the cells, you can trace the nature of the cancer to disorders of growth and disorders of the cell division cycle. And that's going to be our next topic, is how do cells divide? You'll realize if an individual cell has a certain lifespan, when it dies, it has to be replaced. And that is what happens in our bodies the whole time. Our organs are maintained by an orderly set of cell divisions, which continually replace cells as they reach the end of their lifespan. Okay, so we're going to just talk a little bit about cell division and how and why it occurs. I explained, cells need to divide the whole time. And we begin our lives, for example, as a single cell, as a fertilized ovum. Well, from that fertilized ovum, we need to derive all of the tissues of the body, all of them. And they are derived through rapid divisions of that ovum into 2, then 4, then 8, 16, 32 cells, etc. And then during the process of development, all of the different organs are formed, which perform the functions of the whole organism. But key to all of that is this realization that our cells need to divide, and they need to divide in an orderly, controlled fashion. There is the first type of cell division that we're going to talk about are those processes of cell division that are normal for growth, for development, and for replacement. So it's growth, as in the ovum producing a fetus. It's growth, the fetus growing, becoming a birthable baby, the baby growing to become an adult. All of those processes, cell division is going to contribute to them. And the maintenance of the organism cell division is going to replace cells as they reach the end of their lifespan, and they need to be replaced. So when that happens, in those processes, growth, development, replacement, when a cell divides, it is essential that daughter cells inherit the identical DNA. Okay. We can think of it, for example, of a mother cell dividing. The mother cell divides into those two daughter cells must have the identical DNA. They must have the same genetic material to work with. When that kind of division, where we have a division that produces cells with identical DNA, that is a process called mitosis. When I say that DNA is identical, what I mean is, first of all, the DNA sequence is exactly the same. That's one. And second, that each cell has the same amount of DNA. In our cells, we have 46 chromosomes. And when our cells divide, each daughter cell will have 46 chromosomes. The identical number, the identical DNA sequence. That is key and very important. Now, there's a second kind of cell division. And that is the cell division which is associated with reproduction. So, for example, if we produce eggs and we produce sperm, I've told you, we have 46 chromosomes in our normal cells. If I produce sperm with 46 chromosomes and I produce eggs with 46 chromosomes, and the sperm fertilizes the egg, how many chromosomes do I end up with? 92. Okay, well, might work. But that organism with 92 chromosomes in all of its cells, because all of its cells are going to now inherit 92 chromosomes, that's going to now need to produce eggs and sperm. If they produce eggs and sperm that each have 92 chromosomes, we're pretty soon going to end up with way more chromosomes than are possibly viable. So that system is not going to work at all. Instead, when we produce eggs and when we produce sperm, they have half the number. They have half the number of chromosomes, half the amount of DNA. So egg has 23 chromosomes. Sperm has 23 chromosomes. Egg is fertilized by sperm. How many do we have? 23 plus 23, 46 in the fertilized ovum. And that now goes on to divide by mitosis to produce the baby, the fetus. In production of gametes, that is in the production of eggs and of sperm, we halve the amount of DNA. Various other really important things happen, but that's the most important thing for you to remember for the moment. There is one difference between the two processes. This process of division for growth, etc., is called mitosis. The kind of division for production of eggs and sperm for the gametes is called meiosis. And in mitosis, we produce two identical cells. In meiosis, we'll see that we actually produce 4 cells, and they have half the number of the chromosomes. We'll see how that happens in a minute, only in broad detail. I do want to explain to you a couple of things about how our DNA is organized. Again, we have 46 chromosomes, 23 pairs of chromosomes. We say there are pairs of chromosomes because 23 come from your father, 23 come from your mother, the paternal and maternal chromosomes. Now, normally inside the cell, those chromosomes, they exist. This is a single, very, 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 very long piece of DNA. It's one single long molecule of DNA. Same here, one very long molecule. They are in pairs because both the paternal and the maternal share the same genes. So if, for example, we was looking at some gene, a gene for eye color, for example, looked at the paternal, it'll be there, in the maternal, it'll be in exactly the same place, but the genes aren't necessarily the same. One may be for blue, the other may be for brown eyes. But the genes, the same gene is on the same place on the 2 chromosomes. So you'll realize this chromosome will have a whole lot of genes on it for all different things, completely unrelated things, can be on the same chromosome. But when I look at the matching gene, I would see the same genes in the same place, but not necessarily the same genes. They're not necessarily identical. We say they are homologous. So genes for the same thing, but they may code slightly differently. So normally this is how the cell inherits the DNA. Again, we have 23 of these pairs in human beings. Other animals have different numbers. But then during one of the phases of the cell's life, it actually duplicates the DNA. So this molecule here is identical to that molecule. So this one is identical to that. This one is identical to that. They double up. And you may well have seen this as a representation of the kind of X-shaped chromosomes. That's when they've gone through the duplication and they look like this. Otherwise, they look like this, as we'll see in a second. So they duplicate during one particular phase of the cell cycle. It's the longest phase when the cell is doing all its normal business. The chromosomes duplicate in this fashion. So here are our own chromosomes. This is a beautiful preparation of the chromosomes. What they've done is they've stained the different chromosomes. This is before they have duplicated up. So they appear as single. You realize this is a single DNA molecule making up the chromosome. And they've stained the chromosomes different colors. So here, have a look here. This is the normal situation. And that's when the cell is just doing all of its normal business and the chromosomes are all unwound inside the nucleus. You can see the different colors, show you where the rough location of the different chromosomes. Then as cell division approaches, the DNA molecules wind down, they come down and they become short and stocky. So here they are here. And you can see that we can pair them up, in this case by color, by size, and by shape. And there are 23 pairs altogether. 22 of them, the 2 chromosomes look very, very similar, or if not identical to one another. The 23rd pair here are special. These are called the sex chromosomes. And it's because this chromosome pair determines the sex. If we have 2 chromosomes which are identical to one another, like this, those are X, called X chromosomes. And that would be a female. So females have 2 X chromosomes. In males, there's one long chromosome like this, and then the other one is a little short. It wouldn't even be that. It'd be like that arm there, little tiny short stubby chromosome, and that is called the Y chromosome. And the Y, if you have a Y chromosome, then you're genetically male. Have the X chromosomes, you are genetically female. They get 23 pairs, 22 of them, pretty much identical, and then 1 pair of sex chromosomes. So somehow or other, those chromosomes and the DNA that they contain have got to be managed during the process of cell division. And I'm going to go through the stages of that cell division. We'll first of all cover mitosis. That is division normally associated with development, replacement, etc., where we end up with two cells with identical DNA. So this is just a simplified version. This is a simple organism. This could be an organism. There are organisms that just have two pairs of chromosomes. But this shows you the different stages. And in the first stage, you remember that the nucleus is inside its own membrane. And during the first stage of division, that membrane begins to break down because we've got to liberate the chromosomes into the cytoplasm for anything to happen. The other thing we notice in this stage here, we'll notice that the chromosomes begin to condense down. They begin to condense and become visible as visible chromosomes. So this is an organism with two chromosomes. It's got two pairs. There's the one of the one pair, and here's the other of the other pair, one long pair and one short pair there. In the next stage is called metaphase, and we can see different, it's early and then full metaphase, etc. But in early metaphase, we notice something happening inside the cell. And that is, we see that the cell begins to make these tubules here. And those tubules connect to the chromosomes. Here they are here. If you look carefully on this photomicrograph, you can see they originate there, tubules going there and connecting to the chromosomes. So we call this a spindle, and you can just see it beginning to form there. In true metaphase, when the metaphase is full, we can see the spindle very, very clearly. And here it is here, and here. And here are the chromosomes. And what happens is that these chromosomes, once they're tied up to these microtubules, they will move back and forth. They move back and forth and back and forth and back and forth until they're all exactly lined up in the middle of the cell. And that is called the metaphase plate. And as soon as as that happens, the two halves of the chromosomes, get that in your head, the two halves of each chromosome separate from one another. This half go that way, that half go that way. Here they are here. Now wait, let's go back 1 second and have a look. What did we say about the structure of the chromosome? We said that this half of the chromosome is identical to that half. This half of the chromosome is identical to that half. So if I take those two halves and I separate them from one another, I've got the identical DNA sequence on that side and the identical DNA sequence on that side. And I've got the same number of chromosomes, 1, 2, 3, 4, 1, 2, 3, 4, right? So in other words, I've separated identical genome to that side, identical genome to that side. How did I do that? I did it by having identical halves to each chromosome throughout, and then separating the identical halves. Now the cell can divide there. This phase here, where they are separated from one another like that, it's called anaphase. The final phase is called telophase. And telophase is where we restore the old order. We remake the nuclear membrane here around the nucleus. We remake that nuclear membrane, and the chromosomes unwind and carry on their normal business. The whole time this is going on, by the way, the normal business of the cell is held up. The cell can't be doing its normal metabolism and everything else because the chromosomes are all wound up. You can't access the genes. So it's important for the cell to get through this as quickly as possible. And the cell usually does this quite rapidly. They go through this kind of cell division as fast as possible in order to be able to carry on the normal functioning of the cell. And what I end up with two cells which are identical to the original mother cell. Same DNA, same number of chromosomes. During their own interphase, which is the longest phase of the cell cycle, they will double up these two halves of the chromosome, ready for the next division. Those two halves, by the way, we call the chromatids. The whole thing is, when an individual and unique like this, it's a chromosome. And for much of the cell cycle, the chromosome consists of two chromatids. So that's mitosis. Remember the phases: prophase, early metaphase, metaphase, anaphase, telophase, and the cell will now go its normal lifespan into interphase, which is its normal lifespan. Okay, so that should be clear. There is a movie for you to watch. Please make sure that you do so. It'll help you to understand. But now we need to talk about how it is that we can produce sex cells. And this is the process of meiosis. And in some ways, it has similarities to mitosis. In other ways, it is quite different. And it has spectacular results, which I'll explain to you. It's not that simple a process. I'll point out to you the differences between meiosis and mitosis as we go along. So first of all, remember, this kind of division is only going to take place when we produce gametes. So it is going to take place in the ovary, and it's going to take place in the testis during production of egg and of sperm. And it's a very specialized kind of cell division, because in egg and sperm, we need to halve the amount of DNA. We need to cut the chromosome number down from 46, which is our normal number, to 23. And so most of the key events take place at the beginning of the cell division. Now, this all looks very complicated, but in fact, I'll explain to you first of all, in meiosis, there are two sets of divisions. We go through one set here, which we call meiosis 1, and then we go through another set here, which we call meiosis 2. And you see the same phases: prophase, metaphase, anaphase, telophase. They're given numbers for where they occur. So this is prophase 1, metaphase 1, anaphase 1, telophase 1. This is 2. Okay, prophase 2, metaphase 2, anaphase 2, telophase 2. Tells you which set of divisions these are associated with. Almost the most important part of meiosis happens right at the very, very beginning. And I'll explain why I say it's the most important part in a while. You remember that in mitosis, at the end of all the divisions, our chromosomes on either side of the cell division, this cell here, this cell, they were identical to one another, right? Because we separated these two chromatids from one another. We looked at our chromosome, they had two identical chromatids. We separated those identical chromatids apart from one another in mitosis, so that our cells end up being identical to one another. That doesn't happen in meiosis, because here at the beginning here, something very strange happens during prophase. Now in meiosis, the maternal and the paternal chromosomes, which in mitosis behave completely independently of one another, in meiosis, they actually get together and they stick together really, really closely, like this. And they actually break and they swap over bits between the chromosomes. So just think about this. What is happening is, here we've got maternal chromosomes and we've got paternal chromosomes, and they have genes that are at the same place, homologous, but those genes can be different from one another, right? We said eye color gene, they code for blue and maternal, brown in paternal, okay? Now just realize that those chromosomes have many, many, many genes of all different, they're not necessarily related in function to one another, but there are many, 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 many genes governing all sorts of aspects. So the maternal has a certain gene sequence. Paternal will have the same gene sequence, but they may be coding for different things. And what happens in prophase of meiosis is that those chromosomes, the maternal and the paternal, stick close together, and all down at randomly, they may break and swap over information. And what we end up with are chromosomes with entirely new combinations of genes. They're for the same thing, but as I said, the one may have been for blue eyes, and the maternal, brown eyes and paternal, those can swap over, so that now brown eyes is on the maternal, and it's now combined with other genes. We end up creating entirely new sequences of and combinations of genes on the chromosomes. This is one of the most important aspects of this kind of reproduction, because it introduces a huge amount of variability. We take all of those genes, we got 23 pairs of chromosomes with all of their genes, and we're going to mix and match to produce new combinations. Okay, so that's what happens during prophase 1 of meiosis. Is we have this, this chromosomes stick together, and they swap over portions. Have a look here, and you'll see they've shown it diagrammatically here, where before we had just blue, dark blue, light blue. We've now got a chromosome with a dark blue portion and the rest is light blue. This one correspondingly, they swapped there. They've got light blue and dark blue. So this is an entirely new chromosome here. And what happens now is different to what happens in mitosis. Now, when we look, we'll see that now whole chromosomes in pairs, maternal and paternal, line up at that metaphase plate. This is metaphase 1 of meiosis. The chromosomes now are lining up. The entire paired chromosomes line up. Now, in anaphase, entire chromosomes are taken, one to one side and one to the other. Okay, look what's happened. Look what's happened. What did we have here? 1, 2, 3, 4 chromosomes in the original cell. Metaphase, we've still got 1, 2, 3, 4 chromosomes. But look, anaphase, we've got 1, 2, 1, 2. Why is that different to mitosis? Because in mitosis, we split the chromatids apart. Now in meiosis 1, we're splitting the whole chromosomes apart. And immediately we do that, we've halved the number of chromosomes. 2 on that side, 2 on that side. And look, this chromatid here is unique. That chromatid there is unique. It's a unique combination. It did not occur in the parents either. That combination of genes on that chromosome did not occur in either parent. It's a result of crossing over, and this is only one. We may have many crossing overs on both chromosomes, just simplicity's sake, so it's easy to understand. We can produce, just in this we can produce an enormous new variability, a new set of combinations of genes on those chromatids. Now, realize we've now got 2 cells here that we've produced with half the number of chromosomes. Here they are here. there's the one, and there's the other. Each of these cells now proceeds through a division which looks just like mitosis. So here it is again. Here's the metaphase plate, just as in mitosis, the chromosomes line up. And now in anaphase 2, we split the chromatids apart here. And each of those now goes on to form a cell. So from one cell, we've produced 4 cells, 1, 2, 3, 4. And these ones here are unique. These two here in this diagram are unique because they've swapped over bits. They've acquired new combinations of genes on those chromatids. These crossings over are very, very frequent, and all sorts of other things that I won't discuss in this set of divisions contributes to the fact that we're going to end up with usually with absolutely unique cells here. These cells are not going to be identical to one. They are unique. 4 unique sperm cells. In the egg, we're going to end up with a unique egg. It will have combinations of genes that nobody else has ever had. The 4 sperm, they each have combinations of genes that no one else has ever had. And when we put the two together, egg and sperm, we end up with an absolutely unique individual. I forget the exact figures, but the chances of 2 fetuses ending up with exactly the same genome independently of one another, let's say one in Czechoslovakia and one in Peru, ending up being identical, is something that I believe it's something like 10 to the 20, 1 in 10 to the 20, but more. That number of people haven't ever lived on Earth. Each human being is unique. Each combination of egg and sperm is absolutely unique because most of the sperm are unique, most eggs are unique, together doubly unique. And it's all because of this. Everything that happens here in meiosis 1, these are the key events that produce that enormous variability. Okay, so here just summary for you. If you loaded these notes early, please just check because I added this summary. And these are the key events of the meiosis. Be aware that I'm surely going to ask you if you understand differences between meiosis and mitosis. Sorry, there is a little. There is a video for you to watch for meiosis as well, so please make sure you do it. I just want to use this as a kind of a summary because there is one other thing I need to tell you about the processes of meiosis in human beings, and that is it's slightly different in production of an egg and production of sperm. And production of sperm is really easy to understand. Here are our stages of meiosis. We've got this is meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1. And then meiosis 2, each of these cells now divides. Here it goes here. And so we end up at the end of meiosis with 4 cells. In the male, all 4 survive and can form sperm. So that is easy to understand. Here they are. Those sperm each have 23 pairs of chromosomes, and we say that they are haploid. Haploid means halved. The normal situation, all of our rest of our cells, they are diploid, have 46 chromosomes. Sperm have 23. Now in the egg, in the production of the egg, it's a little bit different. Here we call that the original cell an oocyte. Here it goes here. Look at the end of meiosis 1. This is telophase 1. And look what happens. We get a very uneven distribution of cell material. We have one large cell and one small cell that result from telophase in the female. That little tiny body there we call a polar body, and we're going to watch what happens to it. In meiosis 2, we again get division, and here we have again, here's this is going to divide into two, this here will divide in two. This again will divide unevenly there and produce a third little polar body. These polar bodies die. They do not survive. Only this one survives. So from process of meiosis in the female, we only produce one cell. We produce one ovum. Why? Why waste all those other cells? Well, the answer is that the egg has to have a storage of nutrients and everything to supply the growing embryo. And so these divisions are uneven, so that we end up with one large ovum, which contains most of the available cell material, so that when it's if it's fertilized, its initial stages are funded by all of the cell material. And it does so by this uneven division and keeping everything in one big cell. Sperm don't have to do that. Okay, there's a movie for you to watch on meiosis. Please do so. And this is just for fun. If you want to watch it, you can. It's just kind of it's an animation of the whole cell, and you'll maybe recognize some cell activities. Especially, you'll see the motor proteins marching along, dragging things behind them, all of that. But it's just for fun. Don't expect you to remember anything. Okay, so that is that. And then don't forget, on Wednesday, we will just have the quiz, and that quiz will be administered during the set class time. Okay, you must please be ready to take it during the set class time. And I thank you, and I will see you again later sometime. Thank